Hello, everyone, and a very warm welcome to our online audience. Thank you for joining us for this Science AAA's webinar, Deciphering Neurodegeneration, Inflammation, Immune Response, and Alzheimer's. My name is Sean Sanders, and I'm Senior Editor for Custom Publishing at Science. Today, we will bring you the first in a brand new four-part deciphering series, a follow-up to our very popular Deciphering Cancer series from 2017. which, if you'd like to watch it, you can find at webinar.sciencemag.org. Today, we will address the topic of deciphering neurodegeneration. There is a growing recognition that the nervous and immune systems interact under both healthy and diseased conditions. This offers an excellent opportunity to define the role and molecular characteristics of neuroinflammation in neurodegenerative disorders. Chronic activation of the innate immune system is now well established as an underlying factor contributing to neurodegeneration, including Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, and others. A better understanding of how immune responses regulate neuronal homeostasis and of the circumstances leading to dysregulation in pathological conditions is essential to developing effective therapies and mitigating disease impact. In this webinar, our speakers will share their research on immune response-mediated onset of neurodegenerative diseases and explain the genetic and physiological regulation of microglial function in both healthy and diseased states. We have two exceptional speakers with us today who will be sharing their research and expertise. Dr. Beth Stevens from Children's Hospital in Boston, Massachusetts, and Dr. Todd Goldie from the University of Florida in Gainesville, Florida. A very warm welcome to both of you, and thanks for being with us. Before we get started, I'd like to share some information for our online viewers. At the top right of your screen, you'll find photographs of today's speakers and a View Bio link, which you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentations, we will have a short Q&A session during which we will address some of the questions submitted by our live online viewers. So if you're joining us live, please start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology for sponsoring today's webinar. Now I'd like to introduce our first speaker, Dr. Beth Stevens. Dr. Stevens is an associate professor at Harvard Medical School in the F.M. Kirby Neurobiology Center at Boston Children's Hospital and an institute member of the Broad Institute's Stanley Center for Psychiatric Research. Her research seeks to understand the mechanisms regulating the disappearance of synapses by focusing on how immune-related molecules mediate this process, including the role that microglial cells and their connectivity play in neurodevelopment and neuropsychiatric disorders. In addition, her work is providing novel insights into the mechanisms by which immune molecules regulate synaptic and cognitive dysfunction in neurodegenerative diseases, including Alzheimer's disease, insights that could lead to new therapies and biomarkers. Welcome, Dr. Stevens, and thank you so much for being with us today.
Well, thank you very much, Sean, for the invitation and the opportunity to tell you about our work, and also to the listeners for being here today. So, it's become increasingly clear that a hallmark of many brain diseases, including Alzheimer's disease, but also a host of neurodegenerative and other brain disorders, involves the loss of synapses, the junction between neurons, the point of communication between neurons. And synapse loss also happens during normal aging and correlates with cognitive decline and memory impairment. So the big question is, what makes synapses vulnerable in disease and in normal aging? There could be many potential mechanisms, but could there be a common mechanism that underlies this vulnerability? And that's one of the questions that my lab's been intensely interested in understanding. And I feel like we come at this from a unique perspective. I'm actually a developmental neurobiologist by training, and I've spent much of my career studying how synapses are lost during normal development, In fact, synapse loss is a normal process, which you'll hear more about in a moment. But it's become increasingly clear, though, that synapse loss is actually happening in the context of Alzheimer's disease at a very early stage, in this preclinical stage of Alzheimer's disease. It's become clear both from neuroanatomical evidence from human brains and other evidence that this is happening even in the mild cognitive impairment or the mild and moderate stages of Alzheimer's disease. You can see here from that gold line. And so, this raises the question, what mediates this early synapse loss? And obviously this is not a point where we're necessarily having biomarkers to be able to detect such an early stage. And so this is one of the other big gaps, is trying to understand the mechanisms by which synapses become dysfunctional and try to develop new ways of thinking, not only therapeutically, but potentially at thinking about new biomarkers. And we think some of the new evidence that's emerging now in the field might someday lead to some of that insight. So the questions are, what are the mechanisms that drive this early synapse loss in Alzheimer's and other diseases? And of course, ultimately, can we prevent synapse loss and cognitive impairment in disease? So as I mentioned, what we do know is that synapse loss during development is actually a good thing. We've actually learned a lot, at least from my lab's perspective, a lot about how synapse loss happens by studying the developing mouse brain. But we know in humans, for example, synaptic loss and synapse elimination is a normal developmental process by which excess synapses are permanently removed while other connections get strengthened and maintained. This idea of use it or lose it, right? Connections that are meaningful get strengthened while those that are less meaningful get eliminated. And we know from the work of many over the years that this actually doesn't happen all at once, and it doesn't happen at every synapse. There's remarkable specificity, and synapse loss during development happens in distinct critical periods or phases. For example, in sensory systems like the visual system, this happens relatively early, both in humans and in mice. But there are some parts of your brain, like the prefrontal cortex, areas involved in executive function, that continue to prune into early adolescence. This is illustrated here by this Golgi stain image taken from human brain from multiple different individuals over different time periods, where you can hopefully appreciate, at least in these sections here, that there's a remarkable decrease in connectivity between 6 years, for example, and 14 years, showing this elimination of processes and synapses. And we still don't really understand, again, the mechanisms that regulate this process. And so we've been studying this in the context of, because this happens in the mouse brain, we thought if we could dig more deeply into the mechanisms, this might shed new light into fundamental mechanisms of synapse loss that might be relevant to disease. Now, the big question, of course, is, what are those mechanisms? And we know, much of the field for many years is focused on mechanisms that relate to neurons. Of course, neurons are the ones that are being pruned and that are undergoing elimination. But it's become increasingly clear that the resident glial cells in our brains are also actively involved in this remodeling or pruning process, at least in the normal developing brain. And I'm highlighting here on the left sort of just a very oversimplification. So when you think about synapses and you think about neurons, you need to also think about the glial cells. So here we have the astrocytes on the left that both ensheath blood vessels and also synapses. And there's also many types of astrocytes that are also involved in the white matter as well. But for the purpose of this talk, you can see that the processes of astrocytes are not only, we now know from the work of Ben Barres and many others, that astrocytes are critical players not only in helping synapses form, but also helping synapses remodel and prune. And then, of course, on the right is my favorite cell of the moment, which is microglia. And I'm going to tell you a lot more about what we've learned about those cells. But, when I got into this, we weren't really thinking about microglia in the context of pruning. Much of what we knew about microglia came from what we knew about them in the context of injury and disease. They're, in fact, our resident immune cells. They make up about 10% of the cells in our brains. And only recently, through some very elegant state mapping studies, have we come to realize that microglia actually enter the brain, the embryonic brain, from the yolk sac very early in development. They actually enter the brain, at least in a mouse, as early as embryonic day 8. And to me, as a developmental neurobiologist, this was really exciting, and that it put microglia in sort of front and center to be potentially regulating a number of aspects of development. And that got us thinking about, what do microglia do in the healthy brain? What do microglia do in the developing brain? And another really important step forward, and a very important characteristic of microglia that I think are quite distinct from other cells of the brain, are that microglia are incredibly dynamic. And what you're looking at here is just an example of microglia cells in the animal, where you can usually use a reporter line in which all the microglia are labeled green. This is a fractalkine receptor mouse, and either through slice or through in vivo imaging, it's been shown, and you can see how dynamic the processes of these microglia are. And in vivo imaging studies by Wenbiao Gan and Axel Nimmeron and others have shown that these microglial cells are constantly surveying the brain environment, the parenchyma, all the time. And that this tool, this ability to be able to watch these microglia really, in many ways, changed the view in the game in terms of thinking about what they might be doing, especially in the healthy brain. So the question, of course, we wondered is, what are they surveying? And of course, we don't yet know all the roles that they do. They have a lot of homeostatic functions. But what we do now appreciate is one of the things they're surveying are synapses. And here is another movie taken by my graduate student, Jamel Wallace, where we've now overlaid a neuron, shown here in red, and all those bumps are the spines, or dendritic spines, or synapses. And what you can hopefully appreciate here is how dynamic the interactions are between the processes of microglial cells in green and the spines of a neuron. And this has pretty much been observed now by many labs, that they're not only are they listening or surveying what's going on in the environment, but they're actually listening to what's happening at the synapse. And they actually, much like neurons, have a host of neurotransmitter receptors and other ways by which they could sense signals that are coming from neurons in synapses, including neurotransmitters, and that could be used to communicate with the microglia. And at the same time, microglia have the ability to release a number of factors that could be then sensed by the neurons, this idea of a two-way communication. So when we started thinking about this, and especially when these imaging studies started coming online, we started thinking about this idea of pruning and brain development. And that leads us to another very important function of microglia that, again, are probably very unique in many ways to the microglia compared to other cell types of the brain, which is that they're very phagocytic. There are resident phagocytes. And I should also say that we now know that astrocytes are also phagocytic, but we can come to that maybe later in the questions. So they're very good at phagocytosing or eating things. And if you put those two things together, it started raising the question in my lab when I started the lab a number of years ago. Dory Schaefer was my first postdoc, and she was very interested in understanding whether or not microglia were remodeling synapses by engulfing them. And so what she observed is that microglia were particularly phagocytic in the developing brain during the windows of pruning and in places in which pruning were known to happen. And one of those places that pruning was known to happen and has been studied extensively is the visual system of the mouse, where it's been a really terrific model system for studying pruning. And what Dory did is she designed experiments where she labeled the synapses either by a tracer or by other ways. And then what she observed, and this is just a representative image to show that microglia, at least in the visual thalamus of a mouse, what I'm showing you now here is a microglia in green, and those red and blue puncta are the endings of retinal axons, or synapses. And what she observed is almost every microglia she surveyed in the visual thalamus during known periods of pruning that were happening, that microglia were engulfing large numbers of these inputs. And quite importantly, they were doing this during this window of pruning, and then they really ramped down after the pruning period was over. So this study was the first to show that microglia are normally engulfing these synapses, and this, of course, raised a number of questions about how it is that they know which synapse to prune and which ones not to prune. Is there any specificity in this process? And I think this is where it started getting really interesting, because on one hand, the less interesting hypothesis might be that microglia are resident phagocytes, and anything that they see or touch around that time are potentially going to be engulfed in a sort of non-specific way. But the more interesting hypothesis that Dory tested was whether there was specificity to this process, to this mechanism, And more specifically, another way of rewording that is if microglia could detect or sense signals that told them which synapses to prune and which ones not to prune. So going over to this right side is a kind of oversimplification of what synaptic pruning might look like in the visual system, for example. If you were to just zoom in on one postsynaptic neuron, and what you can see is all these red and blue inputs that are innervating that neuron, and this is representative of what you might see early in development, where a postsynaptic cell is innervated by lots of inputs, and they're weak in nature, and there's many of them. But over the course of this window of pruning, you can see that some of the inputs, in this case the blue ones, are being eliminated, where subsets of the red ones are getting strengthened and maintained. And one of the things we know regulates this process of pruning is neuronal activity. And the idea is that the inputs that are more successful in firing the postsynaptic cell, in this case the red ones, tend to win the competition at the expense of the less active neighbors, in this case the blue one. So this idea of activity-dependent competition, which is a very fundamental mechanism of synaptic pruning and remodeling, we started wondering if the microglia could sense differences in activity, and if so, was there a preferential engulfment, let's say, of the blue inputs over the red ones? And actually, Dory did that experiment because in a mouse, you can block activity in one eye, and you can label those inputs, for example, blue, and vice versa on the red. And what she showed in a number of different ways was that there was a preferential engulfment of the less active inputs, suggesting there must be some sort of molecular cues that attract microglia to those inputs. And at the same time, we're also thinking about the possibility that they may also be, in concert with the cues that tell microglia to eliminate synapses, they may also be protective cues that tell the microglia not to eliminate, for example, the red inputs. And what we've discovered is one group of molecules that are involved in the elimination part of that story. And this, again, is where I think we're sort of at the interface of neuroimmunology in the sense that when we started thinking about ways this might work, we started thinking about the ways that a macrophage in the peripheral nervous system might eliminate an unwanted cell, for example. And it turns out that in the immune system, there's a group of molecules called complement, which is a part of the innate immune system. These are secreted molecules that are well known and canonically involved in eliminating either a bacterial cell or an apoptotic cell. And it works because there's a group of these molecules that essentially tag that cell that needs to be eliminated. And macrophages, which are the sort of counterparts in the periphery, these are the myeloid cells in the peripheral nervous system that are the phagocytes, they have receptors for complement that recognize complement, and anything that is coded or tagged with complement gets eaten, literally. And so we started thinking about whether or not a similar mechanism might happen in the brain. And it turns out, and this is work that I did in Ben Barris's lab as a postdoc a number of years ago now, where we unexpectedly discovered a role for these immune molecules, complement, in synapse elimination in the brain. And that what we discovered is that many of these complement molecules were expressed in the healthy brain, and some of them, like C1q and C3, for example, were targeting or binding to subsets of synapses, and that led us to hypothesize that maybe this is one way that microglia could be recognizing those synapses. And indeed, what we found is that if you were to block complement receptor 3, for example, or complement receptors on the microglia, this disrupted pruning, so you had lack of pruning or decrease in pruning. And similarly, if you block complement molecules themselves, we also saw a very similar phenotype, at least in the visual system. So if you put these two ideas together, this placed complement as one of the ways by which microglia are recognizing and eliminating synapses, at least during development, which is I'm showing you now kind of an overview on the left side here of what we've learned over the years. And I'm, in the interest of time, not going to go into some of the other studies, but just to say a few things about the way we're thinking about this. So for one thing, much like the immune system, this process of complement-mediated pruning and microglia engulfment is tightly regulated, much like these pathways are in the immune system. And we're only beginning to understand what turns this pathway on, what turns it off, what's regulating expression of these molecules, as well as their function. And we are now, emerging evidence is starting to shed more light onto this. But one important point is that a lot of what I just told you about today is downregulated after these periods of pruning in the visual system, but also in other parts of the brain. And so you can see in the mature brain, microglia become much less phagocytic than they were in development. And I should also note that astrocytes are also part of this story, and that they release signals that also regulate this process of pruning, and they also mature. And so in the mature, healthy brain, a lot of the mechanisms that regulate pruning get turned down, which is what you'd want to have happen if you want to make sure this is regulated properly. But what if it didn't get turned down? What if there was an aberrant regulation of this pathway? What if too much of a good thing happened, which we envision can happen in the context of development, either through genetic or for environmental reasons? You get an overactivation of this pruning pathway. We hypothesize that could lead to an excess or aberrant pruning, which we think may be quite relevant to a number of neurodevelopmental and neuropsychiatric disorders, including autism and schizophrenia. But also, what if this turned back on again in the aged brain or the adult brain? And we started thinking about the possibility that the same mechanism that is a good thing during development on the left-hand side could become reactivated aberrantly and contribute to that synaptic loss in disease. And so we started thinking about ways we might test that. And to support this hypothesis, there was already a lot of evidence that microglia and astrocytes, reactive gliosis is a common hallmark of Alzheimer's disease and many other neurodegenerative diseases. We also know that there's an upregulation of inflammatory and immune molecules, including complement. But I think probably the most compelling and exciting evidence is more recent human genetic evidence that implicates both microglia and some of these immune pathways I just told you about in human Alzheimer's disease. So large-scale genome-wide association studies, or GWAS studies, have identified more than 20 loci linked to Alzheimer's disease, especially late-onset Alzheimer's disease. And quite interestingly, most are either expressed exclusively in microglia or myeloid cells or enriched in these cells. And of course, this raises this question about, what is it about these genes that's contributing to disease risk? And that is really much of what the field has been focused on more recently. Now that the genetics continue to point to microglia, we now have the ability, and we're starting to get the tools to start to manipulate and to study the biology of these genes. And I think really importantly, much like we did in development, I think understanding what these genes and proteins normally do in microglia biology is going to be really critical for understanding what they do in disease. Because I think we're really faced with a number of challenges. So just because you have GWAS data or genetic data pointing to a particular gene and a particular cell, in this case microglia, we still have to figure out, what the underlying biology is. How is it that microglia contribute to disease risk? And from our perspective, since we really are sort of thinking about the synapse and this cognitive impairment, this aspect of the disease, we want to know, could microglia be contributing not only to the inflammation and the pathology that happens over the course of disease, but could they be contributing very early on, and could they be contributing to synaptic and early cognitive impairment? And if so, how? And we also want to just know when microglia start to change and become altered. That is another fundamental question, and that's not clear yet. Now, what we did to get at this question, at least initially, is we wanted to test the hypothesis that I laid out earlier in a few slides ago, whether or not microglia could be contributing to synapse loss and cognitive impairments, at least in animal models, based on some of the same mechanisms that we've been studying in development. And to get at this question, I should say, So-Yan Hong, a very talented postdoc in my lab who carried out and led these studies, what So-Yan did is essentially said, well, let's go into some of the animal models of Alzheimer's disease, the genetic models, in this case, the J20 mouse model, the APP-PS1 mouse model, and others. And she wanted to know, in these models, where we knew that there was synaptic loss that was already published and shown, and we also know, and I think quite intriguingly, that synapse loss, again, just like development, is not happening everywhere. There's actually regions and time in which this happens. What So-Yan did is found that complement and microglia were upregulated, and that complement was targeting these synapses in these animal models. and that microglia, much like development, were aberrantly pruning or engulfing these synapses. And she went on to ask whether blocking this pathway, either genetically, by blocking complement or complement receptors, or through other mechanisms, including a novel C1q blocking antibody through a collaboration with Annexon Biosciences, which is a C1q blocking antibody. In all three cases, using these different manipulations, we could show a protection against synapse loss in these animal models, which is quite exciting. And so this is an example, and again, I'm just highlighting published work, that this shows a developmental mechanism, or a mechanism that we've been studying in terms of its normal biology, becomes aberrantly regulated in these disease models. And I think now we have something to start to think about in terms of whether we could translate what we learned from mouse into human. Could we start to interrogate and design experiments to start to test and see whether that or not some of these mechanisms are also happening in the human brain? And also how to understand microglia's role in human Alzheimer's disease. Now, there are many, of course, exciting aspects to this, but there are also many challenges. And I think one of the challenges, not just in the context of complement, this mechanism I highlighted today, but the fact is microglia are not all created equal. They have diverse roles in development, and they have diverse roles in disease, both beneficial and detrimental. And so how do you deal with that? You can't just block all microglia because you're not going to be able to get at that specificity, which I think is going to be very important. And as illustrated on this first slide, most of what I told you about today highlighted the beneficial roles of microglia, homeostatic roles like remodeling synapses and keeping neurons healthy. But we know that these cells can also transition states and can become activated, and I'm using that word in quotes in the sense that they change state, morphology, and gene expression, and in some cases they can go on to become extraphagocytic and help to clear toxic proteins like amyloid beta. But they can also be detrimental, right? You could get excess pruning, in which we showed in animal models at least can happen, and they can also promote neuroinflammation. And actually, neuroinflammation is good and bad at types of inflammation, so you can see the complexity that we're talking about here. So this got us thinking about one of the ways that we're trying to tackle this question is that we really don't have good markers or biomarkers or ways to measure these state changes. We don't have molecular signatures for a purely good or a bad microglia because there's lots of states in between. But if we did have a way of looking at different microglia functional states and being able to label them, I think that will give us a lot of potential, not only to identify them, but to manipulate them, and even for potential biomarkers. So imagine, if you will, if you were to look at microglia just by morphology, that doesn't necessarily tell you what they're doing, because there's a lot of reasons why a microglia might look different. But imagine if we were able to come up with a set of markers that could distinguish a microglia that was phagocytic versus one that was migrating or proliferating. And so we've been taking our approach in using single-cell sequencing, drop-seq single-cell analysis, where you can actually profile individual microglia from the mouse brain, and now we can do this in human brain, to try to get a better handle of whether there's different states of microglia could be detected using these methods. And work by Tim Hammond in collaboration with Evan McCosco and Steve McCarroll, using DropSeq, a technology they developed, we could do this single-cell analysis, and what it's revealing is that there are lots of different subclusters of microglia that are being identified. And this is really exciting because these clusters are starting to give us molecular handles on what these different states of microglia might represent. And we can even go on and look in some of these clusters and identify where they are in the brain. And we can use in situ hybridization probes, for example, to try to localize some of the most highly enriched genes that represent these different functional clusters. So the idea, and I'm oversimplifying here, this is all ongoing work, is that we might be able to imagine someday soon identifying molecular signatures that tell us about different functional states in microglia that could be relevant not only to development in normal brain, but also to disease. And of course, we're thinking a lot about how to translate some of the work we've learned into new biomarkers, both CSF and PET imaging biomarkers, as well as, of course, the idea that these might also be therapeutic candidates. So I just want to end by saying that I highlighted today work on Alzheimer's disease. This is published in Science and Science Translational Medicine over the last couple of years, But we also work from now other labs, including in frontal temporal dementia and glaucoma and many other diseases. It seems that there may be common mechanisms across multiple disorders, which also is exciting in that it suggests there may be common mechanisms or pathways we might target. So I want to end just by thanking, I tried to thank some of the key people along the way, but this has been a huge collaborative effort between my lab and a lot of really wonderful collaborators at Harvard, the Broad, Children's, and beyond. A lot of the work, as I mentioned, began when I was a postdoc in Ben Barris's lab, and really it was amazing and that he gave me the ability to carry a lot of this work out in my own lab and supported me and continued to support me throughout this period. And I also want to thank funding and my lab. Thanks so much.
Wonderful. Thank you so much, Dr. Stevens. So our next speaker for today is Dr. Todd Goldie. Dr. Goldie is currently Professor of Neuroscience at the University of Florida, where he directs the Evelyn F. and William L. McKnight Brain Institute and the NIH-funded One Florida Alzheimer's Disease Research Center. He is an active advocate for Alzheimer's disease and neurodegenerative disease research at the state, national, and international levels, serving on 2 state boards that provide input regarding Alzheimer's disease initiatives in the state of Florida, and on the National Medical and Scientific Advisory Boards for the Alzheimer's Association, Bright Focus Foundation, and the American Federation for Aging Research. Welcome, Dr. Goldie, and thank you so much for being on the line.
Well, thank you. So I'd just like to thank the audience for joining us for this webinar, Beth, for a wonderful talk. I'm going to take a little different tact. I'm going to show less of my own data and more talk about I've been sort of heavily vested in trying to, in the preclinical phases, try to find new therapies for Alzheimer's disease and other neurodegenerative disorders. As we all know, there's huge unmet medical need here. And given the increasing interest in the role of innate immunity, one of the questions that's, I think, on many people's minds today is, can we harness this, as Dr. Stevens has alluded to treat Alzheimer's disease and other neurodegenerative disorders? So what I'd like to do today is just provide a little historical context on the role of innate immunity and sort of where it came from in Alzheimer's disease, and other neurodegenerative proteinopathies, sort of put it into context of what we think is an incredibly complex neurodegenerative cascade, and then discuss a few of the issues that I see we need to address as a field going forward before we sort of rush to the clinic with immune therapies. We've actually done some of that, and as I'll show, it's not worked so well. So I think from a historical perspective, Dr. Alzheimer's and others who began to study, and really provide the roadmap for our pathological understanding of Alzheimer's disease noted the presence of activated microglia and astrocytes. So in this slide is just something from a picture from my own lab showing an amyloid plaque in blue, the surrounding microglial cells that come and cluster around the plaque, and the reactive astrocytes that are present. But this obviously is a morphologic description and pathological description. But in the absence of plaques and even other pathologies in many, many neurodegenerative diseases, a reactive gliosis has been long known to be present. I think it's also important from the point of view of our context of neurodegenerative proteinopathies. I mean, I think there's reasonable consensus, not unanimous consensus in the field, that many, many different neurodegenerative diseases are caused by the accumulation of proteins in differential conformations. So not only amyloid protein in Alzheimer's disease, but tau protein in many, many different neurodegenerative disorders, synuclein in Parkinson's disease, SOD and ALS, etc. Furthermore, I think it's important to recognize that Alzheimer's disease is a very complex proteinopathy. Really, to have Alzheimer's disease, you not only need the presence of amyloid, but you need tangles and tau. You, of course, need the clinical syndrome of dementia, and you need evidence for neurodegenerative phases in the disease. And that's just illustrated here briefly. But I think one of the challenges we've had as a field is that despite the fact that there's reasonable consensus that these proteinopathies may trigger the disease and somehow lead to, and this I think moves us away, I used to call this neuronal demise, and I think that we realize now that many, many different cell types, all cell types in the brain are probably affected by these disorders. So it leads to cellular and network demise, this eventually leads to widespread or regional brain organ failure. But what happens in between, we don't know as well. And so I have a series of effects here that have been implicated as downstream effects from the proteinopathy. And I'm not going to go through these in any great detail, but to say I don't think as a field we have a great way to order these or to know which is the most important and therefore which of the ones might be the best places to target. Of course, innate immune activation and inflammation is here. I'd also just like to make the point that at some point in the disease cascade, it may be that these secondary events that are driving neurodegeneration are independent of the initial protein aggregate trigger, which would say that maybe if you target the trigger in the setting of actual disease, when you've already had profound neurodegeneration and cellular demise, you're not going to do much. So some of my interest in the innate immune system really is shown visually here. That this is a series of atomic force microscopy or EM pictures, and some of these are amyloids or amyloid proteins that are formed into an oligomer, and some of these are viruses. And I just want to use this to make the point that you can't really tell these apart. They're roughly the same scale. And the idea is that when you take a normal protein and fold it into a protein aggregate, it becomes what we call in the immune field a danger-associated molecular pattern that can activate innate immunity like a virus or bacteria. And indeed, our lab has some unpublished data that just is shown here with RNA-seq, where we take some primary mouse microglia and dump A-beta aggregates on them. And within an hour, you get thousands of changes in immune genes that look pretty similar to what you do when you dump LPS on these. So I think that this is just one piece of evidence, and there have been numerous published lines of evidence showing this. However, I've gone over some of these points, but in terms of the history of innate immunity and inflammation in Alzheimer's disease. But I wanted to talk about a couple things that I think were important from a historical perspective that drove the field down this road and maybe gave us misled us to some degree. One is that there was I've talked about protein aggregates as DAMPs. I've talked about the gliosis. There's long been known that there's been a catalog of acute phase and other innate immune molecules accumulating in the AD brain or being overexpressed. But a lot of the long-standing hypothesis in the field that pro-inflammatory stimuli drove Alzheimer's disease really came from another amyloid, a systemic amyloid known as serum amyloid A amyloidosis, which is in fact driven by systemic inflammation due to the fact that SAA protein dramatically increases upon an inflammatory stimulus. So you don't get systemic amyloidosis without inflammation, but it's due to the fact that there's an increase in the protein that deposits. So I think that drove the field back in the early days when we had less insights into some of the more mechanistic pathways that we do now. There was also in the late 1990s, and that should be NSAID, epidemiologic data showing an association between long-term NSAID use and a reduction in AD risk. But if we go back to that initial data, there was prior data that it was showing that actually it was the underlying condition, for example, rheumatoid arthritis, that also appeared to reduce risk for Alzheimer's disease. And I want to point this out because if you view the epidemiologic data of NSAIDs being protective from Alzheimer's disease, you say anti-inflammatory strategies should be protective. If you actually view the epidemiologic data as the reason that people were taking a nonsteroidal might have been the real protective factor, you come up with the exact opposite, that underlying pro-inflammatory conditions are. And in fact, clinical trials of NSAIDs and other anti-inflammatory agents have actually been relatively ineffective, and in fact, some cases there is evidence that they may have possible detrimental effects. And I don't think we should read too much into those trials except to use this as sort of a cautionary flag for proceeding forward. We've actually coined the term immunoproteostasis to sort of describe this complex interplay that Dr. Stevens alluded to in terms of that the microglia or innate immune activation states can regulate the underlying proteinopathy. The proteinopathy in turn activates microglial cells, and so there's this complex interplay. And it's also clear that innate immune activation can contribute to a degenerative cascade and perhaps even cognitive dysfunction in the absence of a degenerative cascade, such as we see when people who get an infection or sepsis in the elderly. This can be associated with cognitive decline. But in any way, we like this term because it builds off the term that was originally coined by Richard Morimoto and colleagues talking about protein homeostasis, but we believe the immune system, through numerous, I think, examples, has been shown to be able to also regulate, the protein clearance and deposition. And then, as Beth alluded to, it also now meets the John Hardy doctrine that nothing's important in Alzheimer's disease unless it has a genetic link, and there are numerous genetic links, which I'll discuss a little bit in the future, to Alzheimer's disease. I'm not going to go through this in any detail. I've already shown sort of this cascade, but the idea is that alterations in innate immune activation could regulate the protein aggregates themselves, cellular network aggregate or cellular and network demise, and a lot of other things downstream in this cascade, and that's been our interest in this. But also just point out that anytime there's damage in the brain, we also activate the immune system. So I think this could create a feed-forward loop that could also contribute to the processes. I'm going to just skip over this. One of the things that we found that was quite surprising, and this was a completely serendipitous finding that was published in Nature Neuroscience a few years ago, was that an innate immune signal, in this case overexpression of interferon gamma, can lead to direct neuronal loss and degeneration that was highly selective. So in this case, interferon gamma at 3 months of age in just in a wild-type mouse has really no effect on the nigrostriatal tract, but by 5 months of age, it almost completely wipes it out, while the rest of the brain is largely preserved. And again, I just think this goes to as a nice exemplar of the role of the innate immune system could be very complex. In a parallel set of studies, for example, we were able to show that interferon gamma actually reduced amyloid deposition. And indeed, our focus in the field has been largely at looking at the role of innate immunity and activation states in our mouse models of Alzheimer's proteinopathies. And in fact, we've done a large amount of work, and I'm not going to show all of it here, but what we find is that pro-inflammatory cytokines, as opposed to the original hypothesis, almost invariably decrease amyloid deposition, while anti-inflammatory cytokines actually increase it. In this case, IL-10 or IL-4 are shown here that increase amyloid deposition, and the interferon gamma, TNF-alpha, or IL-6 all create a pro-inflammatory state and decrease amyloid deposition. And so I think these data sort of invalidate that long-standing hypothesis in the field that pro-inflammatory stimuli drove amyloid deposition, at least in mice, and they've all been published in many cases. But as I said, Alzheimer's disease is a complex proteinopathy, and there's been some data from other groups suggesting that if something has a positive effect on amyloid, it may have the opposite effect on tau. And although this is unpublished data, this has largely been developed in collaboration with Dr. Paramita Chakrabarty. What we find is that, again, an anti-inflammatory stimulus, IL-10, can drive tau pathology and the neurodegenerative phenotype induced by this in 2 different models. And I think this means that there's a lot of work left for us to do, and we need to empirically define, really evaluate individual targets, not just in an amyloid mouse, but in a tau mouse, or in whatever the intent to treat type of disease background that we're doing, before rushing things to the clinic. I think we'll find that we might maybe be surprised in terms of what we see. So just sort of as I wrap up here, I want to say that the innate immune system is a well-validated but not a well-defined target, I think, in neurodegenerative disorders, and especially in Alzheimer's disease. It's supported by genetic pathology, biomarkers, epidemiologic, and modeling data. And I'm not, Dr. Stevens referred to some of the genetic data around Alzheimer's disease. There's similar data, although not quite as strong, in Parkinson's disease, and there's an emerging role that some of the ALS genes can also impact microglial function. But obviously the pathology that I referred to, there's CSF studies suggesting altered activation states, the epidemiology, and clearly modeling data. But I think one of the questions is we don't know how to manipulate innate immunity in this, and that I think we have to be really careful here, and this is a sort of a joke on the Goldilocks zone in planetary science, where there's a habitable zone near the star. If it gets too hot, you have negative effects. If it gets too cold, you have negative effects. And I think when we talk about innate immunity, we probably have to move it around carefully within this habitable zone, or without otherwise we'll end up with untoward side effects. So how could we teach ourselves how to set the immune thermostat for benefit in Alzheimer's disease? Well, I think we need to let genetics lead here. We can make inference from our animal models, but we really now, with functional variants in immune molecules such as TREM2, PLC-gamma2, and ABI3, we can learn whether these are immune activating or immune inhibiting, or really how they alter immune function. I think it's also going to be important that we assess the immune phenotype in humans with these variants, not just in our mouse models. And because they're immune molecules, we may be able to do so by accessing immune cells in the periphery. I also think we should perhaps revisit epidemiologic data. There are now many different immune modulatory therapies that are being used, and they've probably been used long enough that we could begin to tell whether they have an impact on the incidence of Alzheimer's or other types of dementia. We need more and better biomarker studies, and I think we also need more modeling studies. If we can't, and I think these need to be done carefully, and we need to look at the effects on multiple endpoints in order to say this is a valid immune target. Looking at, for example, just clearance of amyloid is not going to be sufficient. And I have a couple more slides. And I think one of the things that's beginning to emerge, I don't think the data is completely consistent yet. It suggests that TREM2, which is an immune activating molecule, that the AD risk variants are in fact partial loss of function. And if this is the case, I worry that it may not be feasible in the long term to activate the immune system in an elderly population that might suffer side effects. I'm going to skip through this in the interest of time so we could take some questions, but I do just want to raise the point that some of these genes that we've implicated now in Alzheimer's disease, we already know a lot about in other immune diseases. For example, PLC-gamma2 has both a hypermorphic variant associated with one type of autoimmune syndrome, and it also has a loss of function variant that are associated with a different type of autoimmune syndrome. So I think by probing humans and the peripheral immune system, we could, with both AD-associated variants and compare them to these other variants that cause autoimmune disease, we could learn something important that might help us with understanding whether the innate immune system is in fact something that we could target successfully in Alzheimer's disease. And indeed, I'll just finish with questions. Can we harness immunoproteases to treat AD and other neurodegenerative disorders? I think there are lots of opportunities here. Perhaps we could get disease modification at later stages of the disease. We may be able to see efficacy in multiple diseases. We think this is a common pathway that could cause problems, so maybe one immune therapy might not work just in Alzheimer's disease, but perhaps could work in the setting of Parkinson's or other diseases. I think this is an interesting construct, and I don't know if we have any data to speak of this, but perhaps we could get possibility of a cognitive enhancing effect even in the absence of disease modification. And of course, we know that there are lots of targets that are actually truly druggable within the immune system. Of course, there are challenges. Dr. Stevens referred to some of these, the balance between positive and negative effects of innate immune signaling, and these are very complex. Further, they may be contextually dependent, both on the strength and timing of the innate immune signals and when you chose to intervene in those. And perhaps something that's protective in an early phase of the disease may be detrimental at a later stage of disease. And this is, again, something I worry about greatly, this potential for untoward systemic effects in the immune system. Clearly, PLC-gamma2 variants tell us that targeting PLC-gamma2 would have some liabilities depending on the directionality and how strongly you needed to manipulate it. And then finally, this is one that I think all of us who work with mice that are essentially in bubble chambers realize that immunity in our mouse models may be quite different, and there's actually quite a bit of data emerging around this, that when mice are not immunologically primed, their immune system looks different, and obviously humans have, especially those who are in their 5th, 6th, and 7th decade of life, that we would be, our intent to treat population would be at a very different sort of stage in terms of their immunologic priming. And then finally, I think just to reinforce that empiric validation of targets is needed. So I'll stop there, and I think we'll all be happy to take questions now.
Fantastic. Thank you very much, Dr. Goldie, and thanks to both of our speakers for the wonderful presentations. We are now going to move on to some questions submitted from our online viewers. So just a reminder, if you're watching us live, you can still submit your questions by clicking the Ask a Question button below the slide window, typing it into the message box, and then clicking OK. So the first question I'm going to come to Dr. Stevens with. This is actually an amalgamation of a few questions that we've received on a similar topic, and that is, do we know what causes microglia to start or stop eliminating synapses in neurodegenerative disorders? So do we have any sense of how they're regulated?
Yeah, it's a great question. We know more about how that's regulated in development, which is, I think, a situation where we, unlike neurodegeneration, the reason why we can get at that a little bit more easily is that we have models, for example, in the mouse visual system where we know when and where pruning is happening. And so using the models that we've been that I talked about today, we've been looking for signals that are turning on and off the pathway and trying to get at this question of specificity. And in this case, what we've identified is not only are there molecules like complement that are targeting synapses like these eat-me signals, but we've also identified, interestingly, a number of molecules in the immune system that are called don't eat me signals that are essentially the brakes on the system. And one of these is CD47, which is known to be a don't eat me signal, and also sort of a brake on cancer. In the cancer side, it's of the immunotherapy. But in the brain, CD47 we think might be a molecular brake that tells the microglia to stop pruning or not to prune. And if we knock out those molecules, they over-prune, for example. So this raises the intriguing question that maybe there's a group of molecules, in addition to the ones I mentioned, that work together in this way, and that maybe these same molecules may become dysregulated, the don't eat me signals and the eat me signals dysregulated in disease. And this is now something we're testing in animal models, for example. But there's still many questions we don't understand. We don't understand what is the initiating signal that's triggering the upregulation of complement cascade in Alzheimer's or in neurodegeneration in general. What are the triggers that get those microglia to start phagocytosing more? What are those signals that also turn them off? Again, we're just at the beginning of our understanding, but from our perspective, we're constantly taking what we've learned from normal development and then applying those ideas and hypotheses to the disease models as our first step. So stay tuned. We don't have much more in terms of Alzheimer's, but that's the approach we're taking.
Fantastic. And let me stay with you for another quick question. This viewer asks how complement proteins get into the brain, and wonder if the blood-brain barrier doesn't prevent them from penetrating, or are they actually produced within the brain?
Oh, great. I should have actually said that at the beginning. So what we know is that a lot of complement proteins and molecules are expressed in the brain by neurons, glial cells, mostly a lot of glial cells make them, including microglia and astrocytes. But essentially, complement, at least the classical complement cascade, if you look by in situ hybridization or staining, much of the cascade is expressed in the brain. And it's not to say, of course, we know it's highly expressed in the periphery, but at least in the context of healthy brain and normal development, that's not, it's not the peripheral complement that we're talking about. It's the complement that's made in the brain itself. And we have also evidence that in Alzheimer's models, again, when there's no clear evidence of infiltration of immune cells or breach of the blood-brain barrier, that these glial and neuron-specific expression of these genes go up. And for example, microglia make a lot of C1q. In fact, they're highly enriched in C1q, the initiating protein. But C3, for example, is upregulated in astrocytes. So much of what we think is happening is, especially early in the absence of overt inflammation and blood-brain barrier disruption, is a dysregulation of the expression and regulation of, and possibly secretion of these complement molecules from resident brain cells.
This is Todd. I'd just like to add to that, though, that basically one of the, up until C5, most complement molecules are expressed relatively highly in the brain, but the terminal components of the complement cascade C6, 7, 8, 9 really are not expressed at very high levels, yet there is some reasonably compelling pathology data that suggests you can find those terminal components accumulating in senile plaques, amyloid plaques in Alzheimer's disease. So I think that raises an interesting conundrum. You do find those at relatively reasonable levels within the cerebrospinal fluid on proteomic analysis, but I would suggest that their source, based on extensive RNA sequencing data that's available at 80 amp, really shows that they're not likely to be coming from the central nerve, they're either coming from the filtrate from the cerebrospinal fluid, but not from resident cells.
Yeah, that's really interesting. We haven't been studying the downstream membrane attack complex at all. Much of what we've been focused on is the upstream components. But that's a really interesting observation, and it's good that you pointed that out. So you can imagine that there may be especially when it comes to the later stages of disease, where this may be coming from the periphery and then really kind of triggering the full activation of the cascade, where what I talked about today only led to the sort of activation up to the level of C3. So it's very interesting.
Great, fantastic. Dr. Goldie, a quick question for you. Are there any epidemiological studies showing prevalence of Alzheimer's disease in people with autoimmune diseases that you're aware of, or progression of Alzheimer's disease in these patients?
Well, so I think the answer to that is yes. The original observation was done on rheumatoid arthritis, showing it was protective, and that sort of morphed into the NSAID use. But the most convincing data on NSAID use is around the those who took it for more than 2 years, and most people who take an NSAID for more than 2 years are suffering from some sort of, often it's a degenerative, or pro-inflammatory condition. I don't know how much has been done around other. I vaguely recall something around leprosy. And being protective, but I think it's worth revisiting it more from the point of view of the fact that we have, NSAIDs really don't do a lot, truthfully, to the immune system in terms of its functionality, and many of the NSAIDs that are implicated in those epidemiologic studies are not very blood-brain barrier penetrant. So I think it would be worth looking at the effects of our immune modulators that are being used in people. Anti-TNF agents have been around now for long enough that one should be able to at least do some preliminary assessments as to whether these are potentially altering risk for dementia. I mean, obviously people who so the long answer to that is I can't recall all of the epidemiologic data on other autoimmune conditions. There is some genetic data, that tends to point to common loci between Alzheimer's disease, Parkinson's disease, and some autoimmune disorders. I think that the risk associated with them tends to be very different across the different diseases, though. So, and I think it's still early days there.
So we're coming to the end of the webinar, but I wanted to get in just one more question for both of you, and that is, how do you think that the research that you're doing and recent findings are going to impact what seems to be a very high failure rate in clinical trials, particularly for Alzheimer's disease? And one viewer mentions about 400 plus trials that have failed in the last 14 years. Do you have any thoughts on how we might reassess what's being done in the clinic currently? Dr. Stevens, why don't we start with you?
Well, yeah, I think for one, I think there's a real need to go earlier and to start to understand what are the earliest changes that are happening in Alzheimer's. And of course, that's been a problem, and I think that might alone be one of the contributing factors. But also, we have, fortunately, biomarkers for amyloid and now for tau, but we don't really have we don't have very good biomarkers at all. We don't have any for microglia, which we talked as the focus of today. we know are implicated and even causal based on the genetics. Yet we don't really have a strategy or a biomarker to know when microglia are dysfunctional and be able to start to understand what that means to try to guide therapy in that respect. So I think that some of the work that's coming out where we're going to start to look more carefully at the changes in these cells, especially using human brain and samples in addition to the animal models, is going to be really important because I think studying the human microglia is a gap as well, and we're just now starting to get up and running to do that. But imagine if we had ways that in addition to amyloid and tau PET ligands and biomarkers, we also had a set of biomarkers that represented different functional states of microglia that could be used in conjunction, or I'm sure Todd will mention the idea of other immune biomarkers that gives you a better picture of how to target and when to target. So I guess that's my short/long answer to your question.
I would echo. I mean, I think that with each failure that we see, I think we have to ask. We tested a specific aspect of a specific hypothesis, where we just recently tested whether amyloid production inhibitors would work in the setting of people with longstanding pre-existing amyloid deposition. Our preclinical models would have said it's unlikely to work already if we did the right studies in the preclinical models. The amyloid hypothesis or the proteinopathy hypothesis would say prevent those and you'll prevent the downstream cascade. What we've sort of been doing in the field is akin to saying take a statin, you've had 4 heart attacks, let's lower your cholesterol, and that will cure your heart failure. And, we actually spent money in the U.S. on that trial, and it didn't work with statins in the setting of heart failure, at least systolic heart failure. So I think we have to be really cautious, and Dr. Stevens was dead on when she said what we need to be doing is we need to understand the temporality of events better and where the immune system fits in with that. Then not only on that is, many of the agents that were tested in Alzheimer's disease never really engaged the appropriate target that they were intended to. So that wasn't testing any hypothesis. So early on, we need to make sure that our clinical strategies for translation, these are expensive, as I say, shots on goal. I mean, Alzheimer's trials in phase 3, are hundreds of millions of dollars, if not billions of dollars in terms of expense. And we need to be cautious about moving agents through that are unlikely to work, and we need to make better triage decisions early on. So that includes, determining very rigorously when is the right time that the intervention might be likely to show the maximal effect, to early on showing that there's evidence for target engagement. And 3, being really careful, and I think this is the challenge in the immune system, with if we move things early into asymptomatic phases of Alzheimer's disease, those drugs have to be what I've always used the term safe enough. They have to be pretty darn safe. And that is always going to be a challenge unless we could do very, very targeted immune therapies. So I think we need, this is, I think, Beth's work really highlights the complexity and the complex role of the innate immune system just with respect to microglia and neuronal function, let alone, a whole bunch of other things in the brain. And we need to be cautious. And we could, there's a huge unmet medical need, but when pharmaceutical companies start shutting down their whole neuroscience programs because lack of success, this raises, that still means there's a huge unmet medical need there, and so we have to work smarter, more efficiently, and I think also stay the course.
Yeah, I agree.
Fantastic. Well, great points, both of you, and we're going to have to end there, unfortunately. We're a little bit over time, but thank you for staying with us. So it just remains for me to thank our speakers for today, Dr. Beth Stevens from Boston Children's Hospital and Dr. Todd Goldie from University of Florida. Please go to the URL that will be appearing at the bottom of your slide viewer to learn more about resources related to today's discussion, and do look out for more webinars from Science available at webinar.sciencemag.org. In particular, don't miss our next installment in this current series entitled Deciphering Immuno-Oncology, which will be broadcast in April this year. This webinar will be made available to view again as an on-demand presentation within about 48 hours from now. We are interested to know what you thought of the webinar. Please do send us an email at the address now up in your slide view, webinar@AAAS.org. Again, thank you so much to our panel for being with us today, and to Cell Signaling Technology for their kind sponsorship of today's educational seminar. Goodbye.